Welcome back, everyone. I hope you've all had an enjoyable hour speed networking and watching the demo sessions. My name is Kate Robertson, and I'm the Customer Engagement Manager at BICO. It is my pleasure to introduce our final session of the 2021 BICO Partnership Conference, our Modern Approaches to Drug Discovery panel. Leading today's panel is Visikol co-founder and Chief Executive Officer, Dr. Michael Johnson. A quick reminder that you can submit questions to our panelists at any time throughout the session by typing in the Q&A panel to the right of your screen. We will answer audience questions towards the end of the session. With that, Michael, I'll hand it over to you.
Perfect. Well, thank you for the introduction, Kate. It's my privilege to host this panel session today on drug discovery. We have three great speakers for the panel. So to kick it off, just to give you an overview of who they are, it's Dr. Valani, Dr. Yao, and Dr. Karande. And with that, I'm going to call them up to the virtual stage here to give a brief 1 to 2 minute introduction to themselves and their research, and then we'll dive into some questions. Broadly, the focus here will be on drug discovery, and a little more specifically, we're going to dive into cell culture and then also imaging and image analysis as well. So with that, I'd like to call up first Dr. Tom Villani, the Chief Science Officer and co-founder of Visikol, to give a brief introduction. And I'll move over to Dr. Yao and Dr. Karande after that.
Thanks so much, Michael and Kate and Visikol for putting this together. So yeah, as Michael mentioned, I'm the Chief Science Officer and co-founder of Visikol. My background is in medicinal chemistry. I was working at Rutgers where I was studying natural products chemistry, and I came to develop this tissue clearing technique as kind of part of a side project that we started to realize could have some significant impacts on how tissue imaging was conducted. I actually met Michael in grad school, and we kind of decided to form a company that we called Visikol. We launched that in 2016 to commercialize this technology. And through our past 5-year efforts, we've developed a number of techniques to advance drug discovery assays, what with imaging 3D cell culture models with high content screening techniques, advances in histopathological imaging techniques, and the 3D imaging that was really the core focus of our company when we were founded. We've built a pretty large drug discovery operation where we offer contract research services to pharma clients from around the world. Over the past couple of years, we've grown pretty substantially, and we were acquired by BICO earlier this May, where we joined forces to kind of scale up our efforts and offer more compelling and interesting models to the drug discovery efforts for pharma and research.
Excellent. Thank you, Tom. I'll pass over to Dr. Yao.
Hi, thank you, Michael. Hello, everyone. Just like to thank the organizers for sending the invite. It's been a great conference thus far. So my name is Peter. I'm a postdoctoral fellow at the Holland Lab at Johns Hopkins. And just like to share a screen and share a bit of my research summary during grad school. So I started grad school about 2016, where my lab there focuses on genome instability in cancer, how the genomes of cancer cells are really unstable. So here, just like to show you a cell that is in division or mitosis. Here you can see it relies really on one organelle called the centrosome, which sits on the opposite poles of the cell. So as the name suggests, they are the main microtubule organizing centers of a cell, and it allows cells to accurately partition their genome into 2 daughter cells. So you could imagine they're really important. And when I started my PhD, there was a prevailing notion at the time that cancer cells do not really require centrosomes. They have sort of mutated and to become independent on this really important organelle. But during the first time, we realized that not all cancer cells are as such. There are indeed a lot of cancer cells, especially breast cancer cells, which I focus on, that when you remove these organelles using a specific drug that inhibits a key kinase, these cells are left to divide without poles. And as a result, they undergo catastrophic mitosis and they eventually die. And we were able to further narrow down this genetic interaction down to one gene called TRIM37. It's an E3 ligase. What it does is it destroys these backup clouds that come online when you remove the centrosome. And we were able to publish this last year. And indeed, the study has been extended to way more cancer cells. We predict 10% of all cancer cells have an overexpression of this gene. And a back-to-back study with ours have identified the same pathway in neuroblastoma. So this is pretty promising. this program has been taken up by a drug discovery company. And my work now at Hopkins is just to follow up on this, how this gene interacts with its substrates and looking at a broader range of cancer cells and how it can remove them and stop them from dividing. So I'm really excited to be on this panel, and thank you, Michael.
Excellent. With that, I'm going to pass it over to Dr. Karande to do a brief introduction.
Thank you, Michael. Thank you, Kate. Not to sound redundant, but thank you to the entire BICO family. What a fantastic conference you guys have put together. It's been a pleasure to be part of it. Just to quickly introduce myself, my name is Pankaj Karande. I'm an associate professor in chemical and biological engineering at RPI in upstate New York. And for the past 10 plus years, my lab has focused on two key areas, one being drug discovery and drug delivery, where we use a lot of tissue engineered models. And then the second area is indeed tissue engineering, where we make those models. And for the past 8 or 9 years or so, we've been focusing on designing human skin using 3D printing. We started out this effort with some home-brewed printers, which now belong in a museum somewhere. But we've since been working with Selling and using their platforms. Some of the key achievements we've had is showing that you can actually use 3D printing platforms to create a human skin that's physiologically, biologically, anatomically very similar to human skin. And we leverage that work to now create human skin grafts that add more sophistication, more function to what we've known as the simple, plainer, butter on bread kind of a skin model, but making it more sophisticated by adding adnexal structures. We've looked at incorporating vasculature. We've looked at incorporating hair follicles. And of course, there are other adnexal structures, appendages that we are interested in developing. And of course, the goal here is twofold. One is in regenerative medicine, there's a lot of chronic skin injuries and wounds that would benefit from personalized grafts that do well in patients. Right now, there is a real clinical unmet need that we're trying to address. But on the other hand, we also need better models. Animals are one representation, but they're not always the best representation. So we are creating skin models that can be used for screening topical, transdermal, even follicular delivery methods and compounds. So with that, I'll stop and I'll hand it back over to Michael.
Perfect. Well, thank you. That segues perfectly into the first couple questions I had for the panel here. The first of which is around the development of advanced cell culture models. So it fits in perfectly. And the question that I had is, what do you think has led to this sudden gain of traction of advanced cell culture models in the last 5 years? So spheroids, bioprinted constructs, ex vivo tissue slices. About 5-6 years ago, these things were very academic in nature, and now almost every major pharmaceutical and biotech company in the world is using advanced cell culture models in some way to help answer some specific research question, or broadly, lots of different types of research questions. What do you think has led to that rapid acceleration of the use of these models?
Sure.
So, leaders of the group. Yeah, go ahead.
Dr. Grundy, why don't you start, since from the academic perspective, and I can add some of our point of view. And Dr. Yah, I'd be very eager to hear your point of view, especially in terms of cancer.
Certainly. I think the realization that biology doesn't happen in 2 dimensions on a petri dish, but actually happens in 3 dimensions. I think that realization came to us as an academic community, for over 3 decades now. But just knowing that biology happens in 3D was not enough. I think we needed the right tools to be able to investigate, reproduce that biology at the length scales that really conventional tools, simple pipettes and glassware couldn't afford. So I think it was both this realization that biology is more complex, sophisticated, more nuanced, but complemented with the advances that happened in the technological platforms, 3D bioprinting being one of them, for example, organoid models, spheroid models. I think the confluence of both this basic science with applied science really allowed us to ask questions that was very difficult in the past, if not impossible. So I think this investigation, not only at the cell length scale, subcellular length scale, tissue length scale, being able to do that all on a single platform and a single model, I think that's really accelerated the interest and curiosity of both the academic community as well as the industry community.
Mm-hmm. Yeah, I definitely agree. From an industrial side, there's a couple of pressures that have contributed to a great interest from an economic perspective on top of the technological one. And so there's been a raising concern of growing costs of getting a drug through the clinical trial process. Drug prices have been increasing over the last, several decades. It's a wide concern across the culture that, medical costs are increasing, people are living older, diseases are proliferating, et cetera. And all of that has led to a kind of industry-wide soul searching for how do we improve this process? How do we make drugs for a smaller sum? And, how do we get a drug through a clinical trial for less than $1 billion? And it became very clear that a lot of the failures that happen late stage are due to fundamental limitations in animal models just because of their huge difference from human biology, right? Animals are great at modeling many things, but at the end of the day, they're not humans, and you can only model human diseases so far with them. And so I think that had a major contributing factor. On top of that, from a cultural perspective, there's been a huge shift in how the culture, All good? Great. And from a cultural perspective, animal testing has, in the past, was kind of something that society just didn't think about or accepted as a necessary evil. But over the past 20 years, that's shifted dramatically with the rising concern over environmentalism and animal rights. And it's really been something that the industry has done a huge amount of effort in trying to minimize and find better ways to do this kind of testing, especially given the scientific limitations of animal models. And that comes at the exact right time where the technologies had kind of converged to allow these types of studies to be possible. So I definitely agree with Dr. Grande on that. And the economic and social and cultural pressures were just kind of right at that moment. And so, yeah, it's been a very interesting change in the last 10 years. And Dr. Yao?
Yeah, I think I'll just echo the thoughts of Pankaj and Tom. And as a basic biologist, I think the word is penetrance. You want to see the effect that you see on your cell line, that it's in a 2D culture, translate into more advanced cell culture models, which we now know cancer doesn't exist. They exist in the tissue, and they have surrounding tissue, surrounding cells that they interact with. And I think I've experienced working with a group in the ICR of London where they have curated a biobank of patients. So we went to them and say, could you look at your biobank, look at your RNA, look at your DNA, and picked up organoids that display certain key features that we want and test the drugs on them. And as someone in academia, you would want to see your results translate into something in that kind of setting. So it's kind of promising, and it's something that we yearn for as well, aside from the pressures of eventually making it through drug discovery in the industry, I think. Yeah.
Mm-hmm. Excellent. Thank you for that one. The next one I had was more on the characterization side. And really, I think for Dr. Yao, it's probably, resonates best with your group and your work, but what are the challenges associated with characterizing complex and advanced cell culture models and processes such as tracking the mitosis process in vivo? What challenges do you run into? What are the problems that still need to be overcome? And actually addressing that research question that you're looking at as far as imaging or tools or reagents to actually look into that.
So we look at mitosis, which is within a cell cycle, let's say of a 24-hour cell goes through a G1 stage, a S phase where it duplicates its DNA, a G2 phase before M phase mitosis where it divides. And I think mitosis is actually just maybe 1 hour within a 24-hour window, so to speak. So it's catching these really small, time-wise small processes in the grand scale of a cell cycle, I think, which translates into a lot of microscopy time. You do try to image these processes and you try to sit down in a microscope, although we have a lot of time-lapse microscope where you could just set things up and walk away and let it image. But I guess, yes, the resources and the data that you generate, which I think we might talk about, it is a process, but technologically wise, I think it's more technical rather than challenge for biology. We are pretty good at labeling microtubules. We're pretty good at labeling DNA, looking at how when the DNA compacts and when the DNA divides. I think that's really established, but I think one thing in advanced culture, we have tricks in 2D culture where you could synchronize cells at certain phases of the cell cycle. But in 3D, when cells are interacting, and I think it's known that cells do not divide as frequently in 3D as in 2D, maybe a bit slower. So I think these are the challenges that you might face to acquire the number of events that you need to prove your point. So I think in more advanced, maybe in my experience, I can imagine that you might. It's trickier, but it's definitely doable. Yeah.
Excellent.
Yeah, absolutely. From our experience, it's been basically adapting the advanced cell culture models into the standardized pipeline of the drug discovery process. If you're doing, if you're doing any kind of screening, you basically need to be able to automate it. And so you have to have things fit in well plates. They have to be amenable with like ELISA and qPCR and basically the standard toolkit. Luckily for a lot of 3D cell culture models, that is possible. But as you get more advanced and you get more complicated and you want to introduce aspects of flow and multiple organ systems, this is when it becomes a significant challenge. The same kind of challenges you have to address when you're trying to, when you're trying to evaluate the effects on an animal model. You have a whole complexity of factors you need to keep in mind. And that's probably one of the most tricky processes, parsing out exactly where in the stage you need the complex characterization from the simple, and kind of feeding through which tools are available for the specific model that you're actually using. And likewise, whether that model is applicable for screening based on those limitations.
Yeah, I agree both with Tom and Peter. I'll just add, and not to be redundant, but to underscore this context is so important. I think, on one hand, you have the minimally sufficient but simplistic representation, and you're trying to investigate the system, very specific questions. But as we know, cells interact with the surrounding milieu, whether it's the chemicals, whether it's another cells, the stromal cell population, and oftentimes a lot of that biology is driven by this crosstalk, by this communication between different compartments. And so even when you're trying to characterize a compartment that you're interested in, trying to build the physical, biological, chemical context around it is extremely important. So I think, there's a better awareness, there's more awareness that that entire context is useful when characterization is done.
Great. My next question here is a little bit spicy and controversial, I'd say. So this is one of the topics that comes up a lot in this space is, where do you see it being appropriate to use a spheroid versus an organoid versus a bioprinted model during the drug discovery process? And will one of those replace the others? Do they each have their place? How do they fit in? What are the advantages, disadvantages? And where do they fit into the overall process from your point of view?
Yeah, that's a great question. And to kind of take it off from where Dr. Karande had left off, it really depends on which part of the process that you find yourself. So drug discovery has a whole series of steps that are about first understanding which kind of compounds can even have an effect on the target you're trying to interact with. And the later steps are making sure that that actually has the effect in the biological effect that you want. And even further than that is making sure that it retains the biological effect and doesn't introduce a number of other side effects and toxicities and issues that would make it not useful as a drug. And so in that case, advanced isn't always necessarily better. You want something that can best recapitulate what would happen in real life. However, if you have something that's too complex, you won't be able to figure out why your drug isn't working. You know what I mean? You won't be able to figure out if it's an absorption problem or if it's getting metabolized too quickly, or you know what I mean? And so in the earlier phases of the drug discovery process, the why is critical to understand, because you're iteratively improving the drug. So you need to know what the problem was. And so in the really early phases where you're screening thousands and thousands of compounds, probably you want a fairly simple model, the simplest model that you can make that recapitulates the disease and you can have an effect with. As you get to translational science, this is when complexity, variability, and actually modeling those differences and complexities that happen in biology become more critical. Because at the end of the day, if you're going to spend $1 billion on a clinical trial, you want to make sure that it's going to be successful, right? And so it's at that stage where you have worked out most of the mechanical details about how your drug is going to work, or the toxicity, or et cetera. And at this point, you want to know, is it ready for the big show? And so that's where complexity and getting as close as you can to the in vivo environment is critical. And this is often where animal models fall short. Like I mentioned before, animals are not humans. And so certain diseases are extremely difficult to mimic in animals. And that's why we have a reasonably high rate of failure in clinical trials.
Absolutely. I would say, it really becomes a context dependent solution, right? So it's not so much an either or, and I'll take two examples, right? Both that we've worked on in the past. We started out with investigating the effects of drugs on cancers by creating these cancer spheroids, but very quickly realized that cancer is such a multifaceted disease, and Peter probably can speak more intelligently to it, but the angiogenesis plays a huge role. Having vasculature, understanding what happens to the vasculature, because those become the conduits, right, that either get shut off, cut out, or are not functioning properly. So in some cases, you might want to take a spheroid organoid model, but make it more complex by introducing those features that are really necessary to understand the disease pathology. So that's one scenario. Another example that we've worked on recently is we've been interested in introducing hair follicles. And after multiple attempts of just trying to seed the germinating follicle cells in a 3D printed skin, we didn't go anywhere. And so the approach that worked was trying to take these dermal papillary cells, papilla cells, and creating spheroids out of them, allowing them to grow, mature, and then actually print them within a 3D printed skin structure really helped us not only capture the biology, but that structural engineering around that hair follicle bulge. And so sometimes you kind of have this big toolbox and you bring out the tool that is more apt for the solution that you want. So I'm always in the favor of it not being an either or, but kind of trying to figure out how these pieces fit together in addressing the larger question or larger problem at hand.
Yeah, really well said. I definitely agree with that. It's always a kind of mixture, right, of the available tools that will get you to the right problem. A lot of people, try to divide it into camps of either like self-assembly or stem cells or et cetera, but it's always going to be a mixture of these things that yields us the best solutions. You know what I mean? It's really well said.
Peter, any additional thoughts? Yeah, go ahead.
Yeah. I totally agree with both. I think it's you build up as you go. You go from more simple models as you go on. You test more different things. As I think Tom put it nicely, the big show, you of course want to test it in a more complex model. So I've been recently running CRISPR screens, and let's say the human genome has 20,000 genes, and each genes that you want to knock out is additionally targeted by 4 to 5 guide RNAs. So that puts you in a range of 80,000 guide RNAs in your library. And if each representation you want 500 times, that brings you to about 40 million cells. And then you scale up from there. There's no way you can make 40 million organoids. Well, unless you have infinite money, but I think even biotech companies would. And then you come into technical repeat. So I think, it's definitely you start from a simple model and then you take it up. But that being said, there are camps that are more stringent and say, why not just do a screening platform in a more complex system? You'll catch things that you would not otherwise catch. I agree, but it's always a middle ground that you have to achieve.
Perfect.
Agreed.
The next question I have is along those same lines. As a group, Visikol, Mattek, Cell Inc., we're very focused on developing advanced cell culture models and helping to replace animals as part of the drug discovery and development process. And we're making great strides to do that. But I think there's a lot of complex in vivo situations that are tough to model. And I wanted to get your feedback on when do you think animal models will be removed entirely from drug discovery, if ever? Will it be several years, several decades? Will it be a lot of things being replaced in the short term and some that take a really long time to be replaced? What is your viewpoint on that transition and the time period it'll take to actually get there?
Dr. Rane?
Go for it, Tom.
Answer, but I feel like I'd like you guys to share your opinion first.
Sure. I'm hesitant to say that animals will be completely replaced. I know there has been really concerted effort. If you think about some of the DARPA programs like Human on a Chip, right? And the genesis of those was this realization that disease happens in one part of the body, but the body as a whole is orchestrating responses to it, dealing with it, through multiple processes at the same time. So while we stay disease focused, we have to think about the context of the disease in the entire body. And so, we have circulation, we have metabolism, we have exchange of nutrients. How do you simulate all of that? I think we are now at a point where we're getting really, really good at taking each compartment and trying to understand it better and trying to engineer it better, recreate it better. But once we are there, we have to think about how do we get these compartments to talk with each other, the liver, the heart, the lung, the kidney. You have to think about renal clearance, hepatic metabolism, because that's what happens in a human body. So I think we are moving in that direction with the advances that we have, both in terms of understanding the basic biology, but then also creating really robust and nuanced ways of creating those models. But I think before we have truly what would be a human on a chip that recapitulates that entire dynamics of a physiological system, I think is going to be, it's on the horizon, but it's certainly not within reach yet. And that's just my humble opinion.
Yeah, I would definitely have to agree with most of that assessment. And a lot of it from an industrial perspective is that we have, 50, 60 years of history of like toxicological information, like efficacy, pharmacological information, pharmacokinetic data from mice and rats and rabbits. And so if the goal is to eliminate animal testing, then it will take, probably about a decade or two of doing animal testing in conjunction with some kind of advanced models that we've developed in order to make sure that we're seeing the same crossover, you know what I mean? That we're addressing the limitations, but we're not losing any of the value of that as an analytical tool. And a lot of it just has to do with the historical data. We have so much information, so much knowledge about those model animals that it would take a while before we had the same level of knowledge about our synthesized, body on a chip or whatever, and how they respond to different, types of drugs and responses and et cetera. So I think that's something that the industry is eagerly moving towards, but it is a hurdle, and it's going to require kind of organization across the industry to come to an acceptance of what that new model system will really be. And I don't think anybody has an exactly clear answer of what exactly that will be, but it is clear that that's the direction that it's going. And I know that a lot of the large contract research organizations and a lot of the people developing tissue models have toxicity testing in mind. I know that Europe has completely eliminated animal testing for cosmetics. Matec, who's part of the BICO group, has these skin models that are used extensively for studying cosmetics and toxicology. So we can already see that it's trending that direction. But with the issues of toxicity and safety in drug testing and just the huge amount of historical knowledge, it'll just take a little longer before we have that same amount of data that we can apply for pharmaceuticals. Testing something on skin is a little easier because you basically just need to make sure it doesn't go through the skin and cause any damage. When you're actually putting something into somebody's body, you have to do a lot more thorough toxicity testing. That's where you have to really understand how your model is going to respond in a variety of ways. But yeah, I think that it's definitely the direction we're going. I think it'll be inevitable. But exactly what time frame that will be, I think it'll be at least decades.
Yeah, I agree with everyone. I think surprised COVID hasn't been brought up thus far, but I think when I was still a grad student, the building opposite was developing the AstraZeneca vaccine, and they were doing, as a vaccine, you got to introduce it to an animal. I think it's, as like Pankaj has talked about, you need multi-organ interaction to test this kind of efficacy, and to even introduce, the animal has to has a way or route to deliver your vaccine. So I think as of now, we still need animals, but I think it's definitely trending in a direction. But as of now, I don't see how we can yet achieve that without animal testing. Yeah.
I'll add one more comment to this. There's a complementary arm, right? The regulatory agencies, at what point are they going to feel comfortable that the benefit to risk ratio is well addressed? Because right now, it's a very well-established model of going through, clinical efficacy, safety. I'm sure the FDA is going to have a big say in what will the regulatory policies be around what can be accepted that has not been tested in a representative physiological system before it is brought into phase 1 trials. So I'm sure it's going to be a conversation that has to have the multiple stakeholders at the table. But I do agree with Tom. I think the needle is moving in the right direction. Our dependence on animal models, animal studies is getting lower and lower. The question is, does it get infinitesimally small and disappear? I think is anybody's guess.
Absolutely.
Perfect.
Can I just add one point to that, since Tom and Michael, both of you are sort of more facing towards an industry, the pressure to move away from these models, does it stem from just the failure of this big multi-billion programs to translate a drug, or to what extent does it come from human rights groups or animal rights groups rather? That
I would say that that's a great question.
It's the monetary pressure that's most.
Yeah, that's a really good question. I would say that they have an impact, but more of an impact at like perhaps board meetings of saying, well, can we reduce animal models? You know what I mean? But I think you hit the nail on the head with the first part. The real concern is that animal models just aren't that good at the end of the day. We have terrible failure rates in clinical trials. So like, not only is it, that you can make an ethical argument about animal testing, but more importantly, it's clearly not a great solution to the problem of drug translation. And so I think that's been the biggest push over the last decade or two, is just the economics can't continue if drugs are going to fail and cost $1 billion. Like, we need to fail them much sooner than that. And so that's why there's been a huge interest in the pre-regulatory standpoint of how do we make sure that whatever drug we're going to put even into an animal isn't going to fail, or because there's no reason to waste the money on an expensive animal study if you can knock it out earlier with an advanced model. And so I would say that, even if there was that perfect model, you would still be required by law to do animal testing before you could do a clinical trial. So at the end of the day, it's going to be that confluence of, it'll make sense when we get something that is better and when the regulatory framework catches up to it and agrees that it is better.
I echo that sentiment from the business side. So putting the business hat on, mice are very inexpensive from a dollar figure, but like Tom's getting at, having a drug fail in clinical studies is incredibly expensive. So if we're better able to bridge that gap between in vitro and in vivo, we can save a tremendous amount of money and also in the process reduce the use of animals. So we're coming at it from a lot of different angles. It's the same thing with the debate we have about the environment. Of course, we don't want to pollute the environment. No one does. We want to have solutions that are economical and also provide people the solutions they need to actually get their job done, their work done. So that's it's complicated, but I think we all agree we do want to reduce animals. It's just how do we do it and make sure drugs get to market that are safe, efficacious, and improve the process as we go along.
Yeah. I was just going to add, highlighting, I think it would help the industry to highlight that the 3D models are actually better than animal models at giving data, than to focus on more the ethical conundrum, since a lot of the scientists who study this, animal models are the only option for a long time. So getting into an ethical debate is a tricky one, whereas it's a very easy scientific debate to win because there's some clear shortcomings of animal models. I think that helps push the regulators in the right direction. You know what I mean?
Agreed. The next question I was going to combine with an audience question. So the question that I had was around data. We're generating huge amounts of data. So we're generating 3D models instead of 2D models. We're imaging in vivo instead of taking a single picture of a cell culture model. And how does your lab actually process, handle, use data, and make sense of these enormous amounts of data that we're able to generate? And the question from the audience that ties into this is, do you use quantum computing in any way? And where do you see quantum computing fitting in down the road, if anywhere? So I think we'll pass that. Yeah, let's pass that to Peter first. I'd be interested to see what you do with the in vivo live cell imaging data, how you handle it, how you process it, how you actually analyze it, where do you put it, all those types of questions.
I mean, so this is, giving you a very real problem that we face is just service. We have terabytes of data. We're talking about upgrading our service, or cleaning out or clearing up data for members that have left, projects that have that we wound down. And I think it is a problem. I mean, data, it's good. We always want more data, good data, not just rubbish data. But I think it is a problem we face when you generate loads of data. Someone has to sit down. Although I have friends who are doing their PhDs were doing more computer science related, looking at their heart and looking whether it can a robot or AI script replace a cardiologist at looking at heart clots. So that's just one of the things. I think with AI, I think it's definitely possible, but you still need someone to sit down and see if the program that is trained. But I think for us, yeah, data does become cumbersome. While you generate data, you have to sit down and analyze the data and make sure everything makes sense. And I think it would only get better with better scripts and better programs and knowing what we're looking at. But sometimes you just go out fishing, you just create a lot of data and then you look at interesting things. So that is the process that we scientists also are interested about. We just like to go fishing and go look at interesting phenomenon. And yeah, but yeah, data is a problem just in the physical storage point of sense, and the ability to interpret and analyze them and keep them in a meaningful way. I think it's definitely a problem.
Yeah.
Yeah, absolutely. I mean, we have a whole special team just devoted to dealing with that problem at our company. I mean, I've had to learn way more about computer science than I ever thought a chemist should have to know in order to deal with the types of experiments that we deal with on a regular basis. It's just, we quickly went from an era where the limiting factor was like getting the experiments finished to now being able to parallelize hundreds of thousands of experiments in a few days. And then you have, terabytes upon terabytes of data that you could write several dissertations about. And so all of a sudden the problem has kind of shifted to being inundated by information. Luckily, that's not unique to the biotech space. And there's a huge amount of, miniaturization, extremely inexpensive hard drives, extremely fast internet, extremely available open source libraries, computer programmers who are super familiar with data sets and extremely large volumes of data. So like the time is right for these problems to be solved. It's not quite, those problems haven't really caught up into biology yet, but we see it in almost every other industry. Big data is taken a big bite out of whatever they're doing in one way or another, and pharma's headed that direction for sure. That's why you see all this interest in AI and deep learning and et cetera, which will definitely have its place. But to Peter's point, at the end of the day, science is a human business, and you need a human being to interpret, make decisions, and make hypotheses. And then we use computers and AI to help make that process easier. And I think that is definitely going to only continue as we're able to parallelize and scale these things up. Insofar as how quantum computing fits in, that's something I don't have a huge amount of familiarity with. I know that it can be used to increase the performance of certain types of algorithms. So I'd imagine if quantum computing can be applied to like bioinformatics, then it would probably be quite useful in pharmaceutical drug development into the future. But from my understanding is they have extremely limited like kind of prototypes so far. So I'm sure, in the next, half a dozen years or so, we'll be able to see in what place quantum computing will fit into that. But any advances in computing help with information science, in my opinion, but I'm certainly not an expert on quantum computing.
Yeah. I'll qualify that I opine on both of these topics while not being an expert in either one of them. But I do agree that bioinformatics actually has provided us precedence. I mean, 10, 15 years ago, we were still looking at this big data coming at us, and bioinformatics really helped us parse through it, unpack it, help it understand. So I think it's not a new challenge. It's something that we have been progressively dealing with. And any advances in computing, I think, will be helpful. The only thing I know about quantum computing is it's still practically difficult to be able to run your computing at millikelvins. There are some issues on the technology, the hardware side that need to be addressed. But I would say with open arms, the community would welcome any of those advances to help us better understand these complex biological systems.
Fantastic. No, appreciate that feedback on that. The next one I'm going to pull from the audience as well it's an interesting question. So when we think about our labs, there's always that thing that we'd like to do, but we can't do. I know for me, it's always been, I'd love to have a high throughput light sheet microscope where I can image hundreds of samples a day in 3D. Super cool, doesn't exist. What is the killer application, software, hardware, tool that you would love to have in your lab that you don't have today? Is there something that comes to mind? Is there a limitation you have today you can't get past? yeah, I'd love to hear more about those gaps.
That's a really good question. That's a hard one. I guess if like really what I've always wanted is like I always call it the tiny man, right? If we had a tiny little robot probe where I could drive it in and select the cells I wanted and manipulate things like that, that would be, and I know there are people that are working on these little things, like some kind of mechanically motorized mini cell that you could drive and induce changes to on purpose. I've seen a few papers that come out that are working on this with some kind of ways of manipulating some kind of pumps or something like that. That's something that would be extremely interesting to me for probing biology. If you could essentially like drive the immune cells around and have them, cause effects, I think that would be an extremely useful research tool for probing like an otherwise complex system. I don't know. I don't have a good idea of a good like magic instrument, but
I can. This is somewhat more specific and personal to the kind of research that we do. But as I mentioned before, we really want to have some impactful work done in the regenerative medicine space. And one of the challenges right now, even if you think about skin grafts, which are clinically available, but they're not the best. At the end of the day, our body is so good at recognizing what is self, what is foreign. So this ability to make in the lab beforehand a universal type 0 organ system, whether it's for a graft, whether it's for testing drugs, but something that is going to be truly representative, but at the same time can be personalized, again, to ask specific questions about specific genetic makeup. I think CRISPR is allowing us to get there. We're not quite there yet, but if I had a wish list and there was the top item on that wish list, being able to create sort of, universal type 0 grafts by editing immune cells, being able to tune them correctly, that would probably be at the top of my list.
Great. So instead of talking what I want, I think I'll just talk about a recent one that just came. I think that has been making a lot of waves. I think it's AlphaFold, which is a deep mind. What they have done is look through all the protein structures and using AI to just solve structures of proteins that have not been experimentally solved. And I think that's changed my life. You could just go in and key in your protein, look at your protein, look at the 3D, where does it look at, where does your amino acid point out, does it stick out into solvent, where could be an interaction surface for your substrate. And that has been really interesting. And the fact that Google made it open source and everyone can just jump in, and I think it's great. And I think all these things will just continue to come, and it actually came from AI, big data, and it's just, the confluence of technologies coming in. And maybe it does take for a company as huge as Google that has not really a interest to keep them in-house and to make it available to all, that maybe these are the things that I'm excited about, I think.
Perfect. So we have about 2 minutes here. So this will probably be the last question that we have. And this pertains to the clinical translational space. So the question pertains to doing personalized medicine. So if we're able to go ahead and actually take cells out of a person in the hospital, grow them in an in vitro context and test therapeutics on them, when do you think that will be used in mainstream to actually help better personalized treatments for patients? I know there's some labs that are working on it currently, as well as some hospitals. But when will that be mainstream? When will that add value? When will that come to the forefront of treatment approaches, in your opinion?
That's an interesting question. There's certainly a bunch of people working on that now. I guess right now, like for cancer, for example, there's just not a huge amount of different medical treatments option for the cancer, right? And so you only have to do a couple of tests. And right now, the information that you get from the biopsy is usually enough to kind of figure out where you need to go. There are companies that are working on this, including ours. And the idea will be that in the next 10 years, we'll have extremely precise medicines that affect the patients they're intended extremely well, and probably will have no effect on anyone else. And so that's when this type of procedure where we'll kind of grow this like in vitro twin will become critical in order to give the person the right type of drug that will have very few side effects, but have no effect outside of the person that is really intended to work on.
Yeah, I would just add that genetics really helps us understand sort of the nuances of that personal biology, but we also have to understand that epigenetics, which is sort of this next layer of subtlety, right? That's layered on top of just the genetics. That's also very important. And so there's always going to be this delicate balance of how well do we understand a cell, a tissue information that we've extracted from that patient, and how much of it is really showing the cause effect for the disease or the condition that we want to study. So it's definitely helpful, but I would just add my 2 cents there that the epigenetics portion of it is also equally important beyond just the genetics.
Perfect. I'm going to have to call it there as we have hit the end of our timeline here, but really appreciate all the panelists' feedback on these different topics, and really appreciate all the participants calling in and asking questions along the way. So if you have any questions or follow-ups, the contact info for these speakers is in the portal, so please feel free to reach out. And also you can stop by the Visikol booth as well, and we can connect you there. So thank you, everyone. Really appreciate it.
Thank you for having us.
Thank you.
Thanks, everyone.
Wow, what a way to finish up our sessions at the BICO Partnership Conference. Thank you to Michael and our wonderful panelists for that insightful session. I'm not sure if it was intentional, but you actually did a great job at summarizing a lot of the key themes of this conference, such as moving away from animal models. And I think that summarizes well the key issues being discussed in the life sciences industry and the types of forward-thinking experts in this field are embracing. I think I can speak on behalf of all of us here at BICO that we are proud to be supporting you in this journey. So audience, if you missed any sessions, keep an eye on the lobby page for directions on how to access past sessions from our talented lineup of speakers. They covered animal testing, diagnostics and bioautomation, drug discovery, and tissue engineering. So there's a lot of content there for you to get stuck into. Thanks again to all of our speakers for your inspiring talks over the past 2 days. After 2 action-packed days, I hope everyone is feeling passionate about joining the bioconvergence revolution and changing the future of health. We will now hear from BICO co-founder and Chief Technology Officer, Dr. Hector Martinez, for the closing address.